Good morning. So my name is Katie Whalen. I'm a third-year graduate student in the Department of Biochemistry, and this morning I have the great honor of introducing Dr. Jonathan Licht. Dr. Jonathan Licht received his MD from Columbia University. He currently serves as the Joanna Doe Professor of Medicine and the Chief of the Division of Hematology and Oncology at Northwestern University. He also serves as the Associate Director of the Robert H. Lurie Comprehensive Cancer Center at Northwestern University, as well as the Principal Investigator of the Leukemia Society Specialized Center of Excellence Grant. In addition to this, he's the Senior Scientific Leader of the Northwestern University and NCI-funded Physical Sciences Oncology Center. So on top of all those obligations, Dr. Licht has been running a successful lab for over 20 years. His lab studies aberrant transcriptional regulation as a cause of hematological malignancies. They focus on the molecular biology of acute promyleocytic leukemia, as well as the transcriptional mechanisms of the PLZF transcription factor. We're also interested in chromatin changes in gene expression mediated by the SET protein in myeloma. The title of this talk today is Understanding and Targeting Transcription in Hematology.
Thank you do you hear me? Thank you very much for that kind introduction. And I'm delighted to be here. I want to compliment the organizers for your enthusiasm, for the great program you put together, and for the very interesting posters that we saw last evening. I want to compliment all the grad students and postdocs who put up those posters. I really enjoyed visiting with them. So, here we go. So I'd like to give you, I'd like to start off with some background and some vignettes about aberrant gene regulation in hematologic malignancies, and then we'll go into some of our own work. If the computer will cooperate. I have some research support from Epigenetic, and now my computer is quitting because of that. I don't know.
That's a pretty severe conflict.
Yeah, I'd say. I don't know what it's doing. Okay, so we'd like to talk a little bit about epigenetics, and like all of you, when I don't know anything, I go to Wikipedia. And here's the definition. It's a term in biology used to refer to changes in gene expression stable over rounds of cell division and across generations, but do not involve changes in the underlying DNA sequence. Another definition by Adrian Bird is the structural adaptation of chromosomal regions so as to register, signal, or perpetuate altered activity states. So the fact is that we have one genome it's present in all of our cells, but only some genes are in one cell and other genes are on in another cell. How does this happen? It happens by the regulation of chromatin, the DNA protein complex, also including RNA, which is the unit of gene regulation. This was discovered 120 years ago by chemists like people in this room who discovered dyes that could stain cells, and people looked and they saw something that was colorful and densely colored. That was chromatin. Two types of chromatin, euchromatin, less densely packed, and these are expressed genes, and heterochromatin, which are densely packed and not expressed. The principal component of chromatin, of course, is the positively charged histone protein attracted to negatively charged DNA. Now, there are a variety of transcriptional activation and transcriptional repression mechanisms. I'll just touch on these very briefly. One major concept is they're multi-protein machines, often megadaltons in size, that work in a concerted effort to turn on transcription. Here's a model basal transcription unit with an initiation site, the TADA box, upstream elements, which can attract through these tissue-specific transcription factors, as well as interactions with basal components, complexes such as histone acetylation complexes with components such as CBP, P300, and PCAF, the mediator complex, which is a multi-protein complex that decorates the RNA polymerase II and helps guide RNA polymerase II to the proper start sites of transcription. And the SWI-switch remodeling complex, which actually physically moves chromatin out of the way to allow gene transcription to occur. Here are some of the counterparts of turning off gene transcription. These include Syn3A complexes, Syn3A being a major cofactor that binds to many transcription factors and brings them a variety of complexes, including notably histone deacetylases. The co-rest is a different complex but has some of the same histone deacetylases. Histone deacetylases, you can see, are amongst all these complexes. The NERD complex is a chromatin remodeling complex that helps move chromatin closer together, packs it in, doesn't allow it to be transcribed. And the polycomb complex is a prominent complex in gene silencing, which has an important enzymatic activity encoded by the protein EZH2, enhancer of ZES2. And we'll talk about that in a few more moments. The chromatin remodeling machinery actually uses ATP to hydrolyzes ATP to allow DNA to loop around and move around on the nucleosomes seen here. So nucleosomes can slide along the DNA, moving them out of the way of transcription start sites, or in the case of the NERD complex, bringing the chromatin onto promoters and shutting them down. But this is a DNA-dependent process. Also from Wikipedia, the fount of all knowledge, here's the so-called histone code. What's been clear from the work of my colleague David Alice and many others over the past decade is that histones, the chromatin consists of DNA wrapped around a histone octamer, but there are these tails that stick out from the histone octamer which can be modified on multiple residues. Histones could be acetylated, methylated, ubiquilated, phosphorylated, proline isomylated, and other modifications as well. These show the major sites of modification, the enzymes that can modify these sites. So you can see here that there are literally thousands of different combinations of histone modifications. Collectively, these are known as a histone code. The histone code is written by enzymes. For today, I'm going to focus on histone lysine methylation. Histone lysine methylation is histone lysine are very versatile residues. Histone tail lysine residues can be acetylated, methylated, and when they're methylated, they can be mono, di, or trimethylated. And each one of those methylations can mean something different to the cell. This shows that there are evolutionarily conserved classes of histone lysine methyltransferases that can put on marks that are associated with gene activation, such as histone 3 lysine 4 methylation. And as you can see here, that these enzymes are conserved across evolution, and there are abnormalities of these histone methylation marks in disease. Seen over here, histone 3 lysine 4 activation, histone 3 lysine 27 gene repression, and so forth down the line. Now these marks are written. They're histone lysine methylations or acetylations. They're there. Are they just sort of changing the physical chemistry of chromatin? Well, that may be part of it. But more likely, it's that these types of histone modifications are read by specific modules. For example, the chromodomain specifically recognizes histone 3 lysine 9 trimethylation, whereas the WDR5 domain recognizes histone 3 lysine 4 methylation. So when a histone is modified on this residue, histone 3 lysine 9, tends not to be methylated on this residue. So it'll bring in a complex containing the chromodomain proteins. And very often, the histone modifying enzymes themselves have a domain that recognizes the very modification they make. For example, the WDR5 is part of an MLL, mixed lineage leukemia transcription complex. MLL has itself enzymatically makes the H3K4 mark. WDR5 helps attract more molecules to chromatin, allowing the MLL to spread along the chromatin, making histone 3 lysine 4 marks all along the chromatin in a self-reinforcing pattern. So here's some rules, oversimplified rules, but rules nonetheless. Gene activation generally associated with histone acetylation, histone 3 lysine 4 trimethylation, Histone 3, lysine 9, and 27 are not methylated. They tend to be acetylated. Whereas repressed genes, you lose generally histone acetylation. You lose this histone 3 lysine 4 methylation, and you gain K9 and K27 methylation. Now, there are exceptions to these, but this is generally true. Now, you can imagine that this is a very complicated set of processes, have to be well coordinated across time, across space, across development, across differentiation. And it comes then to no surprise that these types of processes can be disrupted in many forms of cancer. Here's a couple examples. Transcription factors can be affected in cancer. For example, MLL fusion proteins. The MLL histone methyltransferases fuse to aberrant partners in many cases acute myeloid leukemia. You can have overexpression transcription factors. For example, the ERG transcription factor, a sequence-specific DNA binding factor, is rearranged and overexpressed in over 85% of cases of prostate cancer. So you can't really study prostate cancer without knowing something about what's going on here. We studied for many years acute promyleocytic leukemia. I'll show you a vignette about this, which is associated with a fusion between the retinoic acid receptor, a critical nuclear receptor that's activated by retinoic acid, fused to a heterologous protein, PML. So these basic transcription factors can be shuffled, deleted, overexpressed, mutated in cancer. Cofactors that bridge between the basic sequence-specific transcription factors and the general machinery can be mutated as well. Recent sequencing of the genome, as discussed by Dr. Weinstock yesterday, in many forms of cancer, including lymphoma, have shown CBP and P300 mutations. This would lead to a deficit of histone acetylation in these cases of lymphoma. How that causes disease, we don't yet know. But it's very striking from the recent genome sequences that have come out over the past 2 years how frequently chromatin regulatory machinery is mutated in human cancer, including mutations in chromatin remodeling factors, such as ISY mutations in rhabdoid tumors, certain types of connective tissue tumors, and the ARID1A mutation, which was initially found, I believe, in ovarian cancer, but was now found almost everywhere. Very important for chromatin remodeling and seems to be almost a ubiquitous mutation. Intriguingly, in a paper in Nature 2 months ago, histone, there's 3 types of histone 3s, histone 3.1, 2, and 3. Histone 3.3 itself is mutated in human cancer. That's the basic chromatin itself. So you have an aberrant nucleosome with aberrant histone 3.3. It can no longer be histone 3 lysine 27 methylated or acetylated. It's that 27 residue that's mutated. So every bit of this machinery can be affected in human cancer. Can we target this? Can we somehow affect these processes for the treatment of human cancer? Well, we're already doing it in the case of acute promyleocytic leukemia. The retinoic acid receptor is a bistable switch. It can be decorated with co-activator or co-repressor proteins. When present on a promoter in the absence of retinoic acid or at low levels of retinoic acid found in our bodies, it oscillates between a co-repressor bound state and a co-activator bound state. And this will control gene expression and sort of act as a rheostat to encourage or discourage the differentiation of fully mature granulocytes from hematopoietic stem cells. So this is really a tightly balanced process. It's interesting that the retinoic acid receptor is not required for white blood cell differentiation, but acts as a volume control. Well, what happens in acute promyleocytic leukemia is the PML-RAR protein is formed due to a chromosomal translocation, and this seems to be stuck in the off state. It's bound to co-repressors even at physiologic levels of retinoic acid. It doesn't oscillate into that activated state. Only when you add pharmacologic doses of retinoic acid does the co-repressor leave, the co-activator come on, and gene transcription ensues. But you need to add 100-fold more retinoic acid, 10 to the minus 6 molar, rather than 10 to the minus 8 molar, to get this reaction to go. But this reaction does go. And acute promyleocytic leukemia is one form of acute leukemia generally found in adults, which is now about 90% curable, in large part due to this type of transcription therapy. I might add, though, you say, oh, this is a great example of bench to bedside. It actually worked the other way around. Empiric trials in Shanghai, China, by Zhu Chen, who's now the health minister of China, and his mentor, Yang Zhu, showed that retinoic acid happened to cure these patients. In the meantime, Janet Rowley found 25 years ago that there's a consistent chromosomal translocation 15-17 in acute promyleocytic leukemia. And several groups knew that the retinoic acid receptor was on chromosome 17. So they put together a retinoic acid response in these patients, but no other leukemia patients. The 15-17, they asked, is the retinoic acid receptor affected? That's how they found it. It was actually bedside to bench research that showed how this worked. But a very interesting translational research nonetheless. Here's another example how you might be able to target transcription in a hematologic malignancy. Diffuse large B-cell lymphoma seen here. This is a normal sets of B cells in a germinal center. B cells being educated, exposed to antigen, proliferating. This is a very privileged site here. This is a site where DNA damage and apoptosis is suppressed. You want that because these B cells are undergoing rapid proliferation and they're mutating their immunoglobulin genes to select for antibody diversity. If you allow DNA damage responses to go on, the cells would die. So they're suppressed in large part by a protein called BCL6, which is aberrantly rearranged and overexpressed in lymphoma. It normally goes on in the germinal center stage and goes off as B cells further differentiate. Here it's stuck in the on position, encouraging the formation of diffuse large B-cell lymphoma. Now, starting over a decade ago, Arne Melnick, when he was a postdoc in my lab, worked with structural biologists at the Ontario Cancer Institute to understand how does BCL6 actually work as a transcription factor. We knew it repressed transcription, and we knew it had very tight binding to a cofactor known as NCOR and a related cofactor known as SMART. We actually co-crystallized BCL6 bound to NCOR and SMART and found it bound to a unique groove around the side and the bottom of a repression domain called the BTB domain. So we thought that if we took this little bit of SMART or NCOR and linked it to a decoy peptide, we might be able to break up this normal co-repressor-repressor interaction, break up BCL6 repressive function, turn back on apoptosis and other processes in the germinal center, and kill lymphoma cells. And that the idea here, the idea here is that BCL6 is decorated by a co-repressor complex, turns off transcription. If you add a peptide that blocks this, then this co-repressor complex falls off and genes are turned back on. So in fact, that all worked exactly as predicted. And here's a simplified diagram showing 4 lymphoma cell lines with high-level expression BCL6, all killed by this specific peptide, but 4 lymphoma cell lines with low or barely detectable BCL6 expression are not affected at all. So this is a way of drugging the so-called undruggable. We actually formed a peptide that's in IND-enabling studies now to treat this form of lymphoma. And Dr. Melnick has gone on to form a small molecule, a 500 Dalton or so molecule that can mimic the peptide and also block BCL6 function as a form of specific therapy for a hematologic malignancy. So the lesson here is we understand some of the basic biology of these types of transcription factors, how they work, their transcriptional mechanisms, solve it at the atomic level. Maybe we can come up with a therapeutic agent. Another process that's frequently abnormal in hematologic and other types of malignancies is DNA methylation. DNA methylation of CpG correlates with gene silencing. The CpG dinucleotide is relatively infrequent in the genome, but common in the oh, here we go. Get another battery for this? Thanks. Is there more batteries for this? Thank you very much. CpGs are present in promoters in the 5' region of many genes. These methylated CpGs can be stably inherited with cell division. This is a form of epigenetic marking, and it's the ultimate epigenetic mark. Once a gene is heavily methylated on CpGs, it tends to be shut off pretty much for good. There's aberrant methylation in cancer. It turns out that repetitive sequences found near the centrosome, found throughout the genome, even places like endogenous retroviruses, these are hypomethylated in cancer. But specific genes are hypermethylated in cancer, and there seems to be some kind of Darwinian selection to hypermethylate those genes that will suppress cell growth. So the genes that are tumor suppressors are methylated, shut off, allowing cell growth to occur. But there's even more. In the past 2 years, it's been found that DNA methyltransferase itself is mutated in a significant fracture, in this case of acute myeloid leukemia, perhaps leading to really global demethylation problems. This methylation can be started, for example, by a transcription factor complex. For example, we found that PLZF binds to histone deacetylases, the histone methyltransferase G9A, which methylates chromatin. And it turns out that methylated chromatin on histone 3 lysine 9 binds a protein called heterochromatin protein 1, which can recognize DNA methyltransferase, which can then methylate DNA. So in this way, a transcription factor can potentially lead to the hypermethylation of DNA. And in fact, methylated DNA is recognized by specific methyl DNA binding proteins, such as methylated binding protein, MBP. This then can bring in histone deacetylases, more molecules of G9A, which methylate histone 3 lysine 9, bring in more HP1, bringing in more DNA methyltransferase, and again, perpetuating the reaction. This is a recurrent theme, as I mentioned before, of self-reinforcing. Once the reaction gets going, it tends to stay going. And DNA methylation is inherited after replication. There's two types of DNA methyltransferases. De novo DNA methyltransferase that takes unmethylated DNA and methylates it. Those are DNA methyltransferases 3A and 3B. 3A being mutated in human cancer, 3B never so far. And there's a methyltransferase that recognizes hemimethylated DNA and remethylates the daughter strand. This is DNMT31. And actually, there's structural biologic basis for this. My colleagues, Dinsha Patel, has actually figured out how these methyltransferases recognize methylated versus hemimethylated DNA, and there's very specific structural mechanisms. So methylation can go awry in cancer. I mentioned many tumors are hypomethylated, but specific genes are hypermethylated. And really, there's been a use of anti-methylation agents in hematologic malignancies, but we've sort of backed into it. I mean, I've known about this work. A lot of this work was done at Mount Sinai, where I was for many years. And again, it's a little bit of a bedside to bench kind of thing. It was known that these agents hypomethylated for many years. It was also known that if you give some of these hypomethylating agents at high dose, it's just conventional chemotherapy. It just kills cells. So the idea was to give these drugs at a lower dose that was well tolerated, and we try to see whether or not this was associated with changes in DNA methylation. Well, the syndrome that we use these types of hypomethylating agents is called the myelodysplastic syndrome, which is a pre-leukemia. Patients have an aberrant clone within the bone marrow that takes over the bone marrow, and it forms blood cell elements quite poorly. The patients end up presenting with low white cells, red cells, and platelets. And in many cases, these patients will progress on to acute leukemia. Now, this shows that there's a whole panel of genes that are aberrantly hypermethylated, shown in red here, compared to normal hematopoietic cells. And if you treat patients with these demethylating agents, you can find that there's quite good demethylation of these genes. However, we found that there's basically no good correlation between specific genes, such as tumor suppressor genes that are hypermethylated in leukemia and MDS. We found patients who responded had no change in methylation, and other patients who had great demethylation and no response whatsoever. So we do not yet know whether or not this demethylation of specific genes is a marker that the drug got into the patient, or actually a mechanism by which the patient responds. But this is in current clinical therapy. We use it routinely for acute myeloid leukemia of the elderly and for patients at all ages with the myelodysplastic syndrome. So are we targeting transcription? Maybe. But are we affecting DNA methylation? Definitely. Is that related to response? I don't know. So these are some of the current, some background to some of the some ways that we're targeting transcription in hematological malignancies. I'd next like to go into a current interest of mine, which is the problem of global chromatin anomalies in lymphoid malignancies particularly. Lymphoid and myeloid, but we've been focusing more on lymphoid. As I said to you, there are mutations in chromatin regulators of all sorts in hematological malignancies. One theme that seems to be a recurring theme is a focus on the histone 3 lysine 27 methylation site. And this is made by the EZH2 protein. It's an enzymatic component of the polycomb repressor complex number 2. It's lost or mutated in many forms of myeloid malignancy, myelodysplastic syndrome and acute myeloid leukemia. And other components of this multi-protein complex can be mutated as well, such as these components. And this is associated in this work from Nature Genetics 2 years ago with loss of the histone 3 lysine 27 mark. These are two leukemic cell lines without EZH2 mutations. These two have EZH2 mutations. This immunoblock shows a significant loss of histone 3 lysine 27 methylation. Now that doesn't mean just one gene here, one gene here, there. That means all across the genome. This must have some very widespread consequences on genome integrity and gene expression. The histone demethylase, UTX, which oxidizes histone methylated residues to using succin, alpha-ketoglutarate as a cofactor, will oxidize and then demethylate the histone methyl mark. UTX is found on the X chromosome, and it's specific for H3K27. UTX is mutated in many forms of hematologic malignancy. In this sort of tantalizing immunoblot that we're following up by mass spec studies, we found that these multiple myeloma cell lines, which represent a late B-cell malignancy, myeloma cells are basically protein factories that make immunoglobulin, and patients have many symptoms due to, in fact, excess immunoglobulin. These patients with these cell lines with UTX mutation tended to have higher K27 levels than these cell lines without the UTX mutations. So now we're really confused. We have diseases where K27 methylation is down, and others when the K27 methylation is up. Why is this occurring? Don't know yet. It gets even more complicated with the finding 2 years ago, again from these exome sequencing, this is discussed by Dr. Weinstock, of recurrent mutations of EZH2 in diffuse large B-cell lymphoma, particularly a type called follicular and germinal center type lymphoma. These were recurrent mutations in almost one single spot and always found in a heterozygous condition. It turns out if you model, this is not the crystal structure of EZH2, but another histone methyltransferase. Turns out the histone tail fits into this groove right here, into this sort of cage. It turns out that there's a tyrosine right here that's mutated in EZH2, creating a lot of space in here. And now, instead of this histone methyltransferase doing mostly mono and dimethylation, it has enough room to do trimethylation. The problem is, as you add a methyl group onto the histone tail, it gets sterically more and more difficult. Okay, so we won't back up, okay. It gets more and more difficult to add more histone methyl marks. This one's got plenty of room, so it becomes a trimethylase. That's not normal. So in fact, if you take a look by either immunoblot or in this case mass spectroscopy seen right here, these two cell lines with the EZH2 mutation have a dramatically increased histone 3 lysine methylation, histone 3 lysine 27 trimethyl content. That's by immunoblot. and this is mass spectroscopy seeing a decrease of K27 dimethylation and an increase in K27 trimethylation. And it's not subtle. It's about a 3-4-fold change. Again, this means that chromatin all throughout the genome is going to be changed. What happens in terms of gene expression and what happens in terms of phenotype. We've modeled this by taking a B-cell cell line, mouse B-cell cell line, and we overexpress either wild-type EZH2 or these two different mutant forms. Now you can overexpress as much wild-type EZH2 as you want in these cells. You can make quite a bit of it, but you do nothing to the K27 trimethylation. So overexpression doesn't cause this. But expressing either of these point mutants found in patients, you get that depletion of K27 dimethylation and an increase in K27 trimethylation. These are other EZH2 components which are unchanged in expression. So does this have a biological correlate? It does, in that these cells don't grow very well in soft agar, but if we add the mutant EZH2, they readily form colonies in soft agar. I don't think I added it today, but we've more recently found that this is associated with a dramatic increase in K27 trimethylation across many genes by the technique known as ChIP-seq, and those genes are almost always repressed, and there's about 1,000 of them. So that also begs another question. We've have this global change in K27 methylation. It's everywhere, but not really everywhere. It seems to be localized to some spots more than others. We don't know the rules for that. And despite the fact we have K27 trimethylation in many, many places, we're only getting, maybe 5–10% of genes affected in expression. We don't know the rules for this. This is not normal transcription. This is aberrant transcription. And the rules that we learned sort of, oh, here's a transcription factor, and it binds a cofactor, and it binds a histone modifier, and it turns on or off a gene. Those are normal rules. These are pathophysiological rules. We're hoping to try to understand what they are so we know what genes and pathways are affected. One thing that's very promising is at the AACR meeting this past week, there were two companies that are on the verge of developing EZH2 inhibitors. And these will soon be available to the research community, I believe, as tool compounds. And so the question is, will those EZH2 inhibitors reverse this colony formation assay? Will it reverse malignant behavior in models as they're developed? So this is yet another way that we can target aberrant transcriptional regulation. In the last third of the talk, I'll talk about epigenetic disorder in multiple myeloma. Multiple myeloma is a malignancy of very late B cells, as I mentioned, and its molecular pathogenesis was quite obscure until I'd say about 12–13 years ago, with discovery by investigators in France and the United States of recurrent chromosomal translocations that affect about 5 or 6 entities, where there are recurrent chromosomal translocations that link the immunoglobulin promoter enhancer to these genes, cyclin D1, D3, CMAF, MAFB, and notably the one we study, MMSET. These other cases probably have they do have point mutations and a variety of other disorders, but these categories here form distinct clinical and pathological subsets of multiple myeloma. So the MMSET also represents a global chromatin anomaly syndrome. MMSET is a large histone methyltransferase protein with many domains involved in chromatin recognition. These PhD, PWW, P domains, HMG domains, PhD domains, we think that these bind to DNA and to chromatin. The specificity of it is yet to be completely figured out. The SET domain is the histone methyltransferase domain. In multiple myeloma, due to the chromosomal rearrangement, MMSET, which is normally expressed in every cell at a low basal level, is overexpressed by we would estimate at least 10-fold. These are myeloma cell lines without the translocation. Here's MMSET. Here's the overexpression with the 414 translocation. You see some different sizes of the protein due to the fact that the immunoglobulin promoter can invade the MMSet gene either 5' to the entire coding sequence or a little bit internal to the 5' end, leading to transcription of making a transcript that will encode a little less than the full-length protein due to cryptic ATGs within the coding sequence. But in all cases, that set domain is maintained. Is this biologically important for the myeloma cells? We and several other groups have shown by shRNA and siRNA that knocking down MMSet expression level will cause these cells to stop growing. They'll undergo apoptosis. They'll undergo G1 arrest. So MMSet is a growth stimulator for these types of myeloma cells. What mark does MMSet make? What is the chromatin? What's its chromatin signature? Using a radioactive histone methyltransferase assay, we take MMSet made in bacteria, in this case recombinant histones, acetylmethionine is the methyl donor. We were able to show that MMSet will catalyze transfer from SAM to histone, seen here. Positive control is G9A, which mentioned briefly, is a histone 3 lysine 9 methyltransferase, and there it is methylating histone 3. At least in vitro, MMSet is more promiscuous, can methylate histone 3 and histone 4. This is a mixture of 3 and 4, and it seems to methylate both of them. How do we figure out what it really does in vivo? For this, we used two systems, one of which was developed by a gentleman named Josh Loring at Johns Hopkins. He very cleverly knocked out the rearranged MMSet allele in multiple myeloma cells by somatic homologous recombination. He inserted a cassette, including stop codons and transcription termination sites, into the rearranged MMSet allele, leading to a drop of MMSet RNA levels, and as I'll show you in a moment, protein levels as well. He also targeted the non-translocated allele, and you can see that the non-translocated allele is expressing a low level of the protein, so that these levels were still high. Here in MMSet knockout cells, MMSet levels are way down, and you can see a deficit of K36 methylation, and again, with our theme of K27, a surfeit of K27 methylation. So this represents another global chromatin abnormality. This complicated slide, don't look at it, look at it again. These are multiple myeloma cell lines from 414 patients. They all have overexpressed MMSet, some different sizes, as I explained to you before, but they all have a surfeit of K36 dimethylation and a deficit of K27 methylation. These are the cell lines without the translocation. They have high K27, low 36, and it's completely switched over here. So again, this is a profound switch in chromatin. I think I'll just skip this part here. Can we reverse this switch? Is MMSet really causing the switch? So we take our knockout cell lines and we added back MMSet wild type or some point mutants. And let me just skip to the data. So this is a knockout cell line that has low K36 and high K27. We add back MMSet. We gain K36, di and tri in this case. We lose K27. This mutant over here is interesting because it seems super active, just like the EZH2 mutant. It does more K36 trimethylation, and you have a deficit of mono and dimethylation, but it'll still get rid of K27 methylation. This one is completely inactive. So these mutants are very useful. We have a super active and an inactive mutant. So then we could ask, is this histone methylation activity important for growth control? So we take our knockout cells and we add back the dead mutant, and the cells grow pretty poorly. If we add back a wild-type MMSet, we stimulate growth. If we add back the superactive mutant, we also stimulate growth. So the histone methyltransferase inactive mutant does not fully stimulate cell growth. To get full stimulation of cell growth, we need the histone methyltransferase activity. But things are not even get a little more complicated. We made one mutant of MMSet in this PhD domain number 4, which has an intermediate phenotype, intermediate growth, and whereas wild-type MMSet, there's the K36 on, K27 off. When you delete PhD domain number 4, you are turning on K36, but you're not completely turning off K27. There's less than over here, but not as little as over there. So there's two processes going on. Actually, by mass spectroscopy analysis, we found that when MMSET's high in a cell, we get both active K36 methylation and active K27 demethylation. And why is that? Because MMSET can actually bind to the demethylase known as Jumanji D3. So we think it tracks it to chromatin, removes the K27 mark, and adds the K36 mark. So we call this HMT's gone wild. So this complex here will demethylate on K27 and methylate on K36, and we have a global switch of chromatin. What does this do in terms of gene expression? Well, we can do gene expression arrays and more recently RNA-seq and look for genes turned on and turned off. And some of the right types of genes are being affected by MMSET. Genes involved in apoptosis, DNA repair, and chromatin packaging and remodeling. And we actually find that when we look at genes regulated by wild-type MMSET, we see some repressed genes, but a lot of genes really at the high end here activated 20 to 60-fold. If we put MMSET without the HMT domain into a cell, we only get 2 to 4-fold regulation. So MMSET really seems to boost transcriptional activation. And here's an example of the MMSET regulated gene. It's an adhesion gene known as JAM2, may be involved in some of the malignant behavior of these cells. When MMSET's knocked down, JAM2 goes down. In the knockout cell, it goes down. If we add back MMSET, JAM2 goes up. The active mutant turns it on. The dead mutant does not. Again, saying that histone methylation is important for gene expression. Now, we've done genome-wide binding for MMSET across the genome, but I want to share with you a caveat about some of these programs that will say where is a protein bound in a promoter. This looked great here. It's bound in introns and intergenic regions. This is a ChIP-seq, and the program identified peaks. But I want to say, have a caveat about peaks that are called by these programs, because when you look at the raw reads here, this is just MMSET bound everywhere. And what we think is happening is we've just sort of filled up the cup so high, and this protein is so chromatin bound, it's just sort of binding everywhere. But we don't know exactly why it turns on only some genes but not others. We still don't know the answer. So we need new rules for aberrant histone code modification. There's a global increase of K36, but only about 10% genes are changed. And some of these genes that are bound by MMSET are actually repressed and not activated. So we still don't know the exact rules here. We think there are MMSET targets relevant to cancer development. Which ones are actually involved by pathogenesis? Here, we've actually triangulated between downloading a number of patient data sets. There are specific patients with the 414 translocation. They have aberrant gene regulation. We found that in our system, these genes that are turned on in those patients are regulated by MMSET in our cellular systems, and we're knocking down those genes systematically in myeloma cell lines to see if they're critical for myeloma cell growth. I'm going to skip this part here and just go to one final thing right here. If it lets me. Can we affect MMSET? Can we make inhibitors of MMSET to target it for hematological malignancy and other cancers? Well, there are histomethyltransferase inhibitors. One's called BIX1294. It was an inhibitor of G9A, but it does have some effect on MMSET here. But it's at the micromolar level. We really need to get to nanomolar compounds. So it's intriguing, though, that this HMT inhibitor killed MMSET positive cells better than MMSET negative cells. So we're motivated to keep on going for this. So we've developed with Milan Merksic, a chemist at Northwestern, a mass spec-based screen for MMSET inhibitors. Here we take a histone tail and we incubate it with MMSET and let this label transfer occur, let methylation occur. But we do it on histone tail substrates, very small, about 10 amino acid peptides, which are then immobilized onto gold spots. So we have this 96 or 384 well gold spotted plate that then goes in to a mass spectrometer, and we can actually detect the methylation shift of one methylation here. A little bit of dimethylation, but mostly monomethylation. So we put this into this high throughput mass spec machine, and we look for this 14 Dalton mass to charge shift. And in our first 10,000, we got about a half dozen hits out of the first 10,000. We've tested about 3 of those. We were able to get fresh batches from the chemical supplier. That's the one thing about this screening. You don't even really want to go back to the original plates. You want to say, they say it's compound X, get a fresh batch of compound X. Because unless you want to start going back to original plates and figuring out what was in there and do analytical chemistry, you have to take it that it was compound X. So the first 3 didn't work out, but we're continuing. So the last thing I'll say in motivation is MMSET is not just affected in myeloma. This is data from Oncology showing that MMSET as well as EZH2 are highly overregulated in prostate cancer. Recent data from Arul Shanayan's group says that EZH2 actually turns on MMSET in many advanced tumors. And our recent data shows that MMSET actually affects, when you put MMSET into prostate cancer, prostate cells, it encourages migration and invasion seen right here in this assay. So actually, some of the effects of EZH2 in prostate cancer may actually be MMSET effects. Hence, targeting MMSET or EZH2 or both in prostate cancer might be a viable therapeutic target. So I'm going to move to conclusions and say that I think I've given you an overview of some of the disorders that occur in histone and DNA methylation in hematological malignancies. K27 is very intriguing because many of the problems, too much K27, too little K27, or K27 itself being mutated is a recurrent theme in cancer. And this is an intriguing activity we're going to be studying for years to come. We think that HMT activity is important for altering cell growth in cancer, and work I couldn't have time to talk about today, it may affect other processes such as DNA repair and replication. And we think that HMT inhibitors may represent a future therapy for many forms of cancer. So I'd like to thank members of my laboratory who did this work, and a variety of collaborators across the country, and our funding organizations seen here. Thank you very much for your attention.
Excellent talk, Dr. Lurie. Thank you very much. If you have time for questions. Eric, can you run up? Yeah.
I'm just thinking about early diagnosis for multiple myeloma. If anybody looks at, like, for instance, MGUS for MMSET or translocations.
MMSET is found in MGUS. The overexpression is found in MGUS, so it may be a very early event. And this remains a very big mystery. MGUS is the prodromal form of, for others who may not know, is the prodromal form of multiple myeloma. Patients, particularly as they get into their 7th and 8th and 9th decades, more and more normal people who are otherwise healthy, have a hyperproliferation of aberrant clone of immunoglobulin-producing cells. So they have an aberrant immunoglobulin production in the bloodstream. Those patients convert to multiple myeloma at the rate of about 1% to 2% per year. What causes that transition, no one knows. One of my colleagues, we just had a forum at this at the AACR meeting, one of my colleagues at the National Cancer Institute is obtaining specimens from patients serially at diagnosis, and patients have agreed to repeat bone marrow biopsies, which is quite brave, to determine what are the genetic changes that are accumulated by things such as exome sequencing as these patients go along, and to see what mutations may occur in addition to MMSET to stimulate the full-blown syndrome, characterized by even higher levels of immunoglobulin producing cell production and tumors within bones and other complications. So it is an early lesion. What causes it to go from early, sort of smoldering to full-blown disease, no one knows. Yeah.
I really enjoyed your talk because you really focused not only what was known but what was unknown. But you focused on sort of chromatin-wide changes in chromatin marks. But what's the balance between gene-specific changes and genome-wide changes in terms of regulation pathology?
Well, I think it depends upon the pathogenic lesion. For example, PML-RAR in acute promyleocytic leukemia, it's thought that, the original thought was that the PML-RAR binds to retinoic acid receptor normal targets and just shuts them off. And it seems at least partially true. But PML-RAR actually binds to a wider complement of sites than the normal retinoid receptor. So it's repressing the normal retinoic acid receptor sites at a local level. So it won't cause a global chromatin change, it'll cause local chromatin changes. But it actually spreads a little bit beyond the usual local changes to others. MLL fusion proteins cause chromatin changes at specific genes. MLL fusions are attracted to specific genes such as the Hox genes. And on those Hox genes you have an increase of histone 3 lysine 79 methylation, not globally but just on those Hox genes, which are big drivers of malignant cell proliferation. If you give histone, the H3K79 methyltransferase inhibitors, you shut off those Hox genes but not other genes, and cell proliferation goes down. So I think it depends upon the inciting lesion. Yes, David?
John, you showed really nice data that if you modify the epigenome, say by overexpression of M%, you can change the phenotype of cells, including prostate cells. But my assumption is that's only in a very small minority of the cells that are transduced. And so my question is there in fact in a larger set of cells induction of apoptosis or some other phenotype? And do you have any thoughts about what might make one cell versus another that's genetically homogeneous, permissive of the transplant phenotype?
That's really an excellent question because we found in fact that it's been very hard in some cases to make stable transductants with MMSET. Cells don't tolerate it. So those were stable clones. So you're asking if we made stable clones and what happened to the ones that didn't survive? And why did some cells survive and why others didn't? That's a great question. Again, something I don't know, but a great question.
All right, if there's no further
Thank you.
questions, please join me in thanking.